Alright guys, so hopefully you got a little bit of a reprieve and you're ready to go again, because here's part 2. So if you want to feel better, part 2 is 20 slides long, including the slide that we're currently on. So in this one, we're going to talk about how we measure population evolution. We know that it happens, but how? Okay? Again, these are just one of those cartoons that I love oh so much. It shows our little kitty cat, and you know how a lot of cats wear like a little cute bell that rings or whatever. And here's this poor little bird that's like, oh, those sweet sounding bells, they just seem to be getting closer. Or is it just me? Well, let's just say that natural selection is about to take place right here, then and there. You're not going to exist for very long. Okay, here we go. Measuring populations. Okay, so just to recap a little bit, we know that evolution is a change in allele frequency, and that it's going to happen within a population. So hypothetically speaking, what conditions would cause allele frequencies to not change? So what are some things we would have to do so that evolution would not take place? Well, we'd have to remove all of the agents of evolutionary change. So the next 5 things are the conditions under which a population would not evolve or change. The very first thing is that you would have to have a very large population size. So no genetic drift is taking place. We're not moving, or there's no bottleneck effect, there's no founder's effect, no one's leaving or anything like that. 2 would be no gene flow in and out. There's no migration. No one's leaving, no one's coming in. 3 would be no mutation, so meaning no genetic change. 4 would be that mating becomes random. Literally, the next person that walks through that door is going to be the mate. No sexual selection is happening. And number 5 would be no natural selection. Everyone has equal fitness. No one is even slightly more fit than the other. We're all on the same page. So these 5 things would have to happen in order for a population to not evolve. Now, if these sound very similar to the 5 agents of evolutionary change, well, it's because it's them, but in the opposite. All right, so let's talk about Hardy-Weinberg equilibrium. So Hardy-Weinberg is not one person. They're actually 2 people, one who was a mathematician and one who was a physician. I always joke around and say this is what happens when physical science people try to become life science people, but you know, that's just my take on the situation. So it is a hypothetical theory, and it speculates that populations do not evolve, that populations preserve or keep their exact allele frequency. And we use it as a model. We use it as our null hypothesis. So in a way, because it never happens, it proves that evolution happens. I hope that makes sense to you. Now, natural populations are rarely, if ever, in Hardy-Weinberg equilibrium, but it is a useful model to measure if forces are acting on a population. So we kind of use it to see if evolutionary change really is taking place. So the theorem states that we need to count alleles. We need to assume that there are 2 alleles, a dominant and a recessive, and that the 2 of them together must add up to 100% or 1. So it gives us the first equation in the theorem, which says that P plus Q equals 1, where P represents our dominant allele and Q represents our recessive allele. We can also, from this theorem, count individuals. And when we do that, we're still counting frequency, but this time we're counting the frequencies of both alleles. So our homozygous dominant allele will become P squared, P times P, whereas our homozygous recessive would become Q squared, Q times Q, and the frequency of our heterozygotes would become 2PQ, which is P times Q plus Q times P. Again, all of the frequencies have to add up to 1 or 100%, so we end up with a second formula that says P squared plus 2PQ plus Q squared equals 1, where p squared represents our homozygous dominants, q squared represents our homozygous recessives, and 2pq represents our heterozygotes. Right, so here, to put it in pictorial format, here we go again. P plus Q equals 1. In this situation, we are only talking about alleles. P squared plus 2pq plus q squared equals 1. In this situation, we're talking about individuals. So whether you are homozygous dominant for a trait, heterozygous for a trait, or homozygous recessive for a trait. So let's look at an example. We have a population of 100 cats. We have 84 that are black and 16 that are white. How many of each genotype will we have? So this is where we have to start off by thinking about how genes work together and how they affect phenotypes. Hopefully you remember from freshman year that especially in terms of color, homozygous dominant and heterozygotes express the same color. They express whichever the dominant allele is. So in this situation, our black color will be dominant and our white color would be recessive. It means that our cats that are homozygous dominant will be black, but our cats that are heterozygous will also be black. So if we start using our equations from this end, we're going to get the wrong answer because we can't separate which ones are dominant versus heterozygotes just yet. So when we work with Hardy-Weinberg, we always start with our recessive traits, and we kind of work backwards. So we know that we have 16 white cats. Now, because the question specifically says how many of each phenotype, that is going to be a clue to tell you what you're starting with. That 16 is going to represent your q squared value. So your total population is 100 cats. Your q squared is going to be equal to 16 over 100, which is 0.16. Now, if this is your squared value, you simply have to take the square root to find q, which would be 0.4. And then since p plus q equals 1, it stands to reason that 1 minus q would equal p. So 1 minus 0.4 would equal 0.6, and 0.6 would be your p-value. So from here, you would square this p-value, that 0.6, to give you 0.36, and then you would multiply your 0.4 times your 0.6 to give you 0.48. And there you would have had your frequencies for homozygous dominant, heterozygous, and homozygous recessive. All right, so let's see if we can take this Hardy-Weinberg principle and apply it to something. A good application point would be sickle cell anemia. It's an inherited mutation, and it happens in a gene that codes for hemoglobin. Hemoglobin is your oxygen-carrying blood protein. It literally sits in the middle of your blood cell. And if you have the recessive allele, this is what your genotype... Whoops, hold on. Okay, if you have the recessive allele, this is what your genotype would look like. Whereas if you have the normal allele, this is what your genotype would look like. Low oxygen levels cause your red blood cells to sickle, to take on this like half moon and very rigid and sharp appearance if you have this condition. It also, sorry about that, results in the breakdown of red blood cells, and it can cause the clogging of very small and tiny blood vessels, especially the ones in like your eyes, and it can also cause damage to organs because all of these little rigid looking pieces are actually very sharp, and as they're going through, like your kidneys for example, they can tear some of that tissue and cause a lot of damage. This kind of anemia is sometimes and often actually very lethal. So just to give you a little background on the condition. Now let's talk about how Hardy-Weinberg applies to all of this. So when we examine the frequency of sickle cell, we realize that there is a high frequency or a high amount of heterozygotes. About 1 in every 5 Central Africans have this heterozygote condition. We specifically talk about Africans because this is a condition that normally affects people of African descent or, who are fully African themselves. this is a quite large number, this 1 in 5, for an allele that has such severe detrimental effects in the homozygote version. About 1 in every 100 Africans have the condition of full-blown sickle cell anemia. We're saying that about 1 in every 5 Central Africans have the heterozygote condition. Now, considering that people who have the full-blown disease usually die before reproductive age, it's kind of astonishing that we have that many heterozygotes. So why is this particular allele maintained at such a high level in African populations? Well, it suggests that somehow there is some kind of advantage towards being heterozygous. There is a selective advantage, something from an environmental standpoint. Well, that advantage has to do with the condition of malaria. So malaria is caused by a single-cell eukaryotic parasite called Plasmodium. This is what Plasmodium looks like, and it spends part of its life cycle in red blood cells. It's transmitted by mosquitoes. So the mosquito bites someone who's infected. Now it has a blood cell or even just the actual parasite within the blood that it carries in its abdomen. When it bites the next person, that parasite can be transmitted from the mosquito into this new host or this new person that it's biting. The Plasmodium is going to go ahead and invade red blood cells, and that's where it's going to replicate and eventually burst those cells open. And then it goes to the liver, where it's going to mature and reproduce, and it just keeps going and going and going. Malaria is very deadly. Lots of people die from it, and it's very prevalent in African countries. What does that have to do with sickle cell anemia, though? Well, it's what we call the heterozygote advantage. In tropical Africa, where malaria is common, if you are homozygous dominant for normal hemoglobin, then you tend to die, or at least you have reduced reproductive abilities from malaria. Those who are homozygous recessive for sickle cell anemia, meaning that they actually have the condition, they tend to die or have reduced reproductive abilities from sickle cell anemia. Well, if you are a heterozygote, you are a carrier. And it means that you're relatively free of both conditions. You're able to survive and reproduce, and this ability makes these carriers more and more common in the population. Being heterozygote gives you some inbred protection, let's say, against malaria, but you also don't have the full-blown condition of sickle cell anemia, so neither of them really affect you. Okay, I just want to draw your attention to these two maps of Africa, and it shows the areas in Africa where the parasite that causes malaria is prevalent, and then this map over here shows the area in Africa where the sickle cell allele is prominent. So we've already discussed relative fitness in our video before the last, the one that talks about Darwin and unique gene pools, but it bears mentioning again. So relative fitness is the contribution of an individual to the gene pool of the next generation as it compares to or is relative to the contribution of other individuals. So that's why when we use phrases like survival of the fittest and the struggle to survive and the struggle for existence, we should not think of them as it relates to actual individuals, but instead as it relates to the species or population as a whole. Okay, so let's delve a little bit deeper into natural selection. There are what we call 3 modes or 3 ways that natural selection can alter the frequency of traits. And the way that it works depends on which phenotype is favored. So in all of these next slides, you're going to be seeing this graph repeated constantly. This blue line is always going to represent the original population, and then you'll have different amounts and colors of mice down at the bottom because the population we're talking about here are mice. So there are 3 kinds or 3 modes of natural selection. They're called directional, disruptive, and stabilizing selection. And we're going to go ahead and step through those now. So starting with directional selection, Directional selection occurs when conditions favor individuals that exhibit one extreme of a phenotypic range. So if we look at our little mice friends down at the bottom, we have really light colored mouse, and it changes all the way up to a really dark colored mouse. Directional selection is going to favor just one of these, either the really dark or the really light. So in this example, we're going to use our dotted line to represent our original population. This arrow represents that this is the point at which the directional selection is going to start working, and you'll notice there is a shift in the population showcasing just the darker of the mice and not the lighter. Okay, now this can go both ways. We can have it feature just the dark mice, but we can also have it feature just the light mice. It depends on the environment in which they're found. So it commonly occurs where the population's environment changes or when members of a population migrate to a new and different habitat. So it just kind of depends on which of these you would find, depending on if the environment has changed or if individuals have moved. The next method is called disruptive or diversifying selection, and it occurs when conditions favor individuals at both extremes. So not just one over the other, but both extremes are favored, and the individuals in the middle are the ones that are no longer fit for that environment. So the intermediates have a lower relative fitness, whereas the really light colored mice and the really dark colored mice are the ones that now have an advantage. So again, dotted line represents our original population. White arrow represents where within that population this selection method is going to take place. And we see that the light colored mice increase in population and the dark colored mice increase in population, but everyone in between no longer exists. Or at least those are the ones that are harmed. Okay, the last one is called stabilizing selection, and it's stabilizing selection removes extreme variance from the population, and it preserves just the intermediate types. So again, dotted line represents original population. White arrows represent where selection is going to take place. So we've gotten rid of our really light mice. We've also gotten rid of our really dark mice. The only ones that remain are the ones that are kind of in the middle, the mixture of those two colors. So this reduces variation. It means that this entire population is going to look, or is going to be made up, of mice that look like this little guy. So it reduces variation, and it tends to maintain the status quo, for lack of a better term, for a particular phenotypic character. So everyone looks like him. We don't have a whole lot of variation in this population. Okay, sexual selection we've talked about already. We're going to touch on again. It's also a form of selective pressure, but it's where individuals with certain inherited characteristics are more likely than other individuals to obtain mates. And it can result in what we call sexual dimorphism, which is where the male of the species looks very different in regards to the female of the species. And that's because it has secondary sexual characteristics. So like, for example, these are pheasants. This would be the female, this would be the male. He has bigger tail feathers and is more brightly colored. Here we have deer, and again, this guy here with the antlers would be the male, and this would be his doe, no antlers, so it's just a way to tell the two sexes apart. So there are two kinds of sexual selection. There is intrasexual and intersexual. Intrasexual selection is selection within the same sex. Individuals of one sex compete directly for mates of the opposite sex. This is what we tend to think of when we think of males exhibiting dominance. So the two males within that species are both fighting for the attention of the female. They're fighting each other in the hopes that one of them will be proven, the weaker of the two, or the least likely to be a potential mate or a good mate, and whoever is the winner is going to get the girl. Okay, versus intersexual selection, which we call mate choice. And it's where the individuals of one sex are choosy. So often these are females, so the females tend to be the ones that are choosy, and they select mates based on their showiness, so how big their mane is, or how many brightly colored things they can bring them, or how well they dance, stuff like that. That would be intersexual selection. Alright, so hopefully this wasn't too painful and you got through it and you're still alive. Feel free to take a really nice long break, and I will see you guys when I see you guys. Bye!